morning everybody. We have been discussing the structure of skeletal muscle, the molecular basis of muscle contraction, and we are aware that basically there are two types of contractile filaments, the actin filaments and the myosin filaments. The myosin filaments are the thicker filaments, they are made up of myosin molecules, and we also studied the structure of myosin molecule. It is a hexapeptide containing 6 polypeptide chains. The 2 chains are heavy chains with a higher molecular weight, and there are 2 chains which are light chains present in the head portion, and the part of the myosin molecule which projects outwards and the head at its tip, that is called as the cross bridge. In case of actin filament, there are 3 types of proteins. There are 2 F-actin strands, or the filamentous actin strands, which are twisted around each other. These strands are formed by polymerization of G-actins, or the globular actins. There are certain ribbon-like proteins called tropomyosin, and this tropomyosin molecule, which is a sort of ribbon-like molecule, in the resting state covers the active site of actin filament and therefore prevents interaction between the actin and myosin. And there is a third protein called as troponin, which has 3 subunits: troponin T, which binds it with the tropomyosin; troponin I, which inhibits the interaction between actin and myosin; and troponin C, which binds with the calcium ions. Now coming to the cross bridge cycling, that is exactly how the muscle shortens. The muscle shortens by sliding of actin filament towards the center of myosin filament, and therefore the length of the sarcomere is reduced, and therefore there is a shortening of the muscle. Now this cross bridge interaction occurs in the form of a cyclical manner. In the resting state, the head of the myosin, the ATP has already bound to it. Binding of ATP itself stimulates the ATPase activity of the myosin head, and therefore the ATP is broken down, and the energy of the ATP is used to keep the head of the myosin molecule perpendicular towards the active site of the actin filament. But please note, there is no interaction because the active site of actin filament is covered by the tropomyosin, and therefore the head of the myosin, even though it has a strong affinity for active site, it cannot bind with it. Now when calcium ions enter the sarcoplasm, the calcium ions bind with the troponin C. As they bind with the troponin C, the tropomyosin these strands which are covering the active site, they move deeper into the groove between the 2 F-actin strands, and as they move deeper, they expose the active site, and therefore the head of the myosin immediately binds with the active site of actin filament. Once this happens, the intramolecular force of the head of the myosin causes bending of the head. As the head bends, it is still in contact with the active site of actin, and therefore as the head bends, it carries the actin filament towards the center of the myosin. Now as it goes through this movement is called as power stroke, because it is through this movement that the actin filament is moving towards the center of myosin filament. Now once the power stroke is completed, the ATP attaches to the head of the myosin, and as ATP attaches, the head of the myosin gets detached. The ATP binding itself stimulates the ATPase activity of myosin head, and therefore the ATP is broken down, and the energy of the ATP is used to bring it back to the perpendicular position, which we call as the recovery stroke. At the end of the recovery stroke, again the head of the myosin is in close contact with the subsequent G-actin molecules. So again it attaches. Again there is a power stroke dragging the filament, detachment, recovery stroke, attachment to the subsequent site, and this cross bridge cycling continues, and therefore there is sliding of actin filaments into the myosin filaments. Right. What causes relaxation of a muscle? When the calcium ions are pumped out of sarcoplasm into the L tubules, the calcium ions from troponin, they dissociate. Now this causes the tropomyosin strands to slide back onto the active sites of actin filament covering them. Thus the heads of myosin filaments cannot attach to the active sites, the cross bridge cycling stops, and the process of contraction ends. Right. Now relaxation just doesn't mean stoppage of contraction. The muscle has to come back to the original length, and this is brought about by the series elastic component which I mentioned last time. I also mentioned that there are the contractile components like the actin and myosin. There are series elastic components, that is the elastic tissue present in the muscle tendon, and there is a parallel elastic component which is in the form of this epimysium, endomysium and so on. So the elastic tissue present in the muscle tendon, which has been stretched during contraction, then pulls out the interdigitated filaments back to their original position, and the muscle relaxes to the resting length. Right. We then start with the excitation, contraction, coupling. Excitation, contraction, coupling. Now what is this? When a muscle is excited by the impulse coming through the neuromuscular junction, it leads to a series of events which ultimately cause contraction of the muscle. Thus the process of excitation of muscle is coupled or leads to the process of contraction. The excitation of muscle brings about release of calcium ions into the sarcoplasm, and then the calcium ions bind with the troponin C and then initiate the process of contraction. So this is called as excitation contraction coupling. If you remember, the neuromuscular transmission causes development of what we call as motor end plate at the neuromuscular junction, and this motor end plate or end plate potential that occurs at the motor end plate causes activation of voltage gated sodium and potassium channels that are present adjacent to the neuromuscular junction. So once the end plate potential reaches a certain threshold value, these channels open and therefore there is beginning of action potential, and this action potential is then transmitted along the sarcolemma on either sides of the neuromuscular junction. Now the basis of excitation contraction coupling is called the sarcotubular system. Sarcotubular system is the basis of excitation contraction coupling. The sarcotubular system consists of two parts. One is the sarcoplasmic reticulum, L tubules, L for longitudinal. So sarcoplasmic reticulum has certain tubules which are arranged in a longitudinal direction, that is along the length of the muscle. So those are called as longitudinal tubules. And number two, there are certain tubules called transverse tubules, as these tubules are situated transverse to the direction of the myofibrils. Right. The sarcoplasmic reticulum. This is an elaborate, modified, smooth endoplasmic reticulum that surrounds and forms sleeve-like structure around each myofibril. So as you can see in this diagram, the blue colored structure here is the sarcoplasmic reticulum, and you can see that these parts of sarcoplasmic reticulum, this is the cross section of a myofibril. So these are all the myofibrils inside a muscle fiber, and here these are the parts of sarcoplasmic reticulum. They act like a sheath, or they form a sort of sheath around each myofibril. So sarcoplasmic reticulum is a membranous anastomotic network, and many of these tubular structures are arranged longitudinally that is parallel to the myofilaments, hence called as L tubules. At either ends, these tubules are dilated and are in close contact with T tubules. These dilated portions are called lateral sacs or terminal cistern. So here you can see this blue color structure is the sarcoplasmic reticulum, and you can see that some parts of this sarcoplasmic reticulum are arranged in the form of these blue colored tubules arranged longitudinally. So these are the L tubules, and you can see that at either ends these L tubules form a dilated structure, as you can see here. You can also see from this side, these are the dilated structures and they are called as terminal cisternae or the lateral sacs. So this is lateral sacs or terminal cisternae on one side, these are the lateral sacs and terminal cisternae on the other side. Right. So that is the orientation of L tubules. There are certain processes arising from the terminal cisternae and extending towards T tubules. They are called as junctional feet. These processes contain certain receptors called Rhinodine receptors. So these are the proteins, or these are the molecules with which the substance rhinodine combines, therefore they are called as Rhinodine receptors. So as you can see here, this is one aspect of the triad. Let us see, these are the L tubules, and towards the one end it forms this dilated sac called as terminal cisternae, and from the terminal cisternae there are these processes extending towards the T tubules, and these are called as the junctional feet, and they contain the receptors called as Rhinodine receptors. The Rhinodine receptors act as calcium release channels. I repeat, the Rhinodine receptors present in the junctional feet or terminal cisternae of L tubules, they act as calcium release channels through which the calcium ions stored in the L tubules are released into the sarcoplasm. So remember, normally in the resting state, the sarcoplasmic calcium concentration is very less. These calcium ions are actually pumped into the L tubules, and therefore the concentration in the sarcoplasm is kept very low. The Rhinodine receptors act as calcium release channels, and therefore when these channels open, there is release of calcium from this terminal cisternae into the sarcoplasm. The calcium ions stored in the L tubules, or the storage of calcium in the L tubules, is facilitated by a calcium binding protein called calsequestrin. So this process helps in storing more and more number of calcium ions by binding to calsequestrin. You also might be knowing about some other calcium binding substances. If you remember, there is something called as calmodulin, which is present in the smooth muscle and many other structures. Then there is something called as calbindin, which is present in the intestinal epithelial cells and so on. So the one that is the calcium binding in the skeletal muscle, which is present in the L tubules, is the calsequestrin. The sarcoplasmic reticulum also contains the calcium pump, which pumps the calcium ions from the sarcoplasm back into the L tubules. Okay. Right. So you can see here, this is the sarcolemma, that is the membrane, cell membrane of the muscle cell. I'll be talking about the T tubules shortly. These are the T tubules, and you can see that these are the L tubules of the sarcoplasmic reticulum, and these L tubules have these projecting portions which we call as junctional feet or terminal cisternae, and in the wall of these terminal cisternae are the rhinodine receptors, and these rhinodine receptors are nothing but the calcium release channels that are present in the terminal cistern. Not only that, in addition to this, the wall of the L tubules also contains the calcium pump. Now this calcium pump, which is present in the L tubules, is activated as soon as the calcium concentration in the sarcoplasm increases, and therefore as the calcium is released through these calcium release channels into the sarcoplasm, automatically the calcium pump or the calcium ATPase which is present in the wall of the L tubules is stimulated, and it starts pumping the calcium ions back into the L tubules. Right. So these are the two important things in the L tubule. One is the calcium release channels called as the rhinodine receptors, and second the calcium ATPase present in the wall of the L tubules, and which pumps out the calcium back into the L tubules and helps in relaxation. Right. So here also you can see sort of diagrammatic representation. This is the sarcoplasmic reticulum L tubule, and this is the calcium release channel, that is the rhinodine receptor, and when this calcium release channel opens, there is a release of calcium ions into the sarcoplasm, and as I said, the presence of protein calsequestrin helps in holding the calcium ions in the L tubules. Right. This is just a simple diagram to give you an idea about a triad. A triad is a complex formed by three structures. So here you can see in the skeletal muscle, this is the T tubule, which I'll be talking about this later. This is the T tubule which is formed by the invagination of sarcolemma, and as this T tubule invaginates into the sarcolemma, it is surrounded by the terminal cisternae of L tubules on either sides. It is surrounded by terminal cisternae of L tubules on either sides, and so this complex consisting of a central T tubule and the two lateral L tubules together forms a triad. So the transvestibules or the T tubules are the inward tubular extensions or the invagination of sarcolemma, that is the cell membrane of the muscle. These T tubules penetrate the muscle fiber transversely all the way from one side of the muscle fiber to the other and surround the myofibrils on their way. Thus they form a transverse grid through which myofibrils penetrate. Now let us see this. So you can see that the yellow colored structures are the T tubules. You can see here, this is the so these are the T tubules, yellow colored structures. They are formed by invagination of sarcolemma, as you can see here, and then as they enter the muscle fiber, as they come across a myofibril, these T tubules divide, and as they divide, they surround the myofibril. Suppose it enters here and this is the one side, then it will divide and the other one will surround from that side, and again they will join together. Then again as they come across another myofibril, again they will divide and then surround the myofibril and then reunite. And in this way they will move, or they will run across the muscle fiber from one side directly to the opposite side. And therefore if you see the pattern of T tubules alone, you will see that the T tubules form a grid or a network. They will form a grid or a network, and this grid will be penetrated longitudinally by the myofibrils. So this is the basic concept of triad. Right. So transverse tubules are the tubular extensions or invaginations of sarcolemma penetrating the muscle fiber transversely all the way from one side to the other, and as they pass they surround the myofibrils on their way. Thus they form a sort of grid or a network which is arranged transversely and through which the myofibrils penetrate. There are two T tubules in each sarcomere. So the diagram will make this very clear. As you can see, this is the one sarcomere. As you can see, this is the one Z line, this is the other Z line or Z disc, and therefore this is a sarcomere. And you can see that there are two T tubules for each sarcomere. So there will be two triads. There will be two triads in each sarcomere. So there are two T tubules in each sarcomere situated at the junction of light and dark band. So here you can see that you can see that the portion containing the thick filaments or the myosin filaments, as you know, it is called as the dark band or A band or anisotropic band, and the portion between two dark bands is called as the light band. So here you can see this is one sarcomere and this is the junction of dark band and a light band here. This is the junction, and at each junction there is one T tubule, and thus there are two triads. There are two triads in each sarcomere in the skeletal muscle. The lumen of the T tubule contains extracellular fluid for the simple reason it is an extension of the cell membrane. So whatever is outside the cell membrane, that is extracellular fluid, it also goes through the T tubules. So the fluid that is present in the T tubules is extracellular fluid. The membrane of the T tubule contains another protein which is called as dihydropyridine receptor. The membrane of the T tubule contains an integral protein which is called as dihydropyridine receptor. So this is showing the invagination of the sarcolemma, and you can see these membrane of the T tubules, where they come in close contact with the L tubules, they contain certain proteins called the dihydropyridine receptors. This protein is a voltage sensitive L-type calcium channel present in groups of 4. So what is this dihydropyridine receptor? It is present in the membrane of the T tubule and it acts as a voltage sensor. It acts as a voltage sensor L-type calcium channel present in groups of 4. It mainly acts as a voltage sensor and is activated when action potential spreading along the sarcolemma dips into the muscle fiber through the T tubule. The principal function of T tubule, therefore, is to carry the excitation process or the action potential into the interior of the muscle fiber, thus activating all the sarcomeres in all the myofibrils rapidly. So, rapid and simultaneous activation of myofibrils is the purpose served by this dihydropyridine receptor. Right. Now going back to the diagram. Yes. So these are the dihydropyridine receptors present in the T tubules. These DHP receptors, they act as voltage sensors. They detect the change in voltage, and as they detect the change in voltage or as there occurs depolarization, these sensors activate the Rhinodine receptors, which are the calcium release channels in the L tubules. So when the action potential goes through the or spreads through the membrane of the T tubule, the DHP receptors, dihydropyridine receptors, are stimulated. They in turn bring about the opening of the calcium release channels, that is the Rhinodine receptors, and then the calcium ions are released in.
Sir.
Right. So, if muscle has to contract, it has to be stimulated. Propagation of electrical current or action potential should spread along the sarcolemma. There should be rise in intracellular calcium, and that will be the final stimulus for contraction. So these series of events linking the excitation with the contraction is called as excitation contraction coupling. The action potential of the skeletal muscle lasts only for a few milliseconds and ends before the contraction occurs. The period between the action potential initiation and the beginning of contraction is called as the latent period. And so, as you might have guessed, excitation contraction coupling occurs within the latent period. Alright, now let us recapitulate the events right from excitation of a neuron to the contraction of muscle. The first event will be generation of an action potential in a motor neuron. So there will be generation of action potential in a motor neuron, number one. This action potential will travel down the axon right up to neuromuscular junction. At the neuromuscular junction, the axon terminal will release the neurotransmitter. The neurotransmitter will bind with the receptors, the nicotinic receptors that are present on the postjunctional membrane. Binding of the acetylcholine with the receptor will open these receptors, which are mainly the channels for the sodium ions, and so the sodium ions will enter the postjunctional membrane and will produce a depolarization, which we call as end plate potential. So end plate potential is a local depolarization that occurs in the postjunctional membrane or the postsynaptic membrane. Now this local depolarization spreads to the adjacent part of the sarcolemma, where there are voltage gated sodium and potassium channels. So the action potential is initiated there at the sarcolemma, adjacent to the neuromuscular junction or adjacent to the motor end plate. And this action potential then spreads along the sarcolemma, dips into the T tubules, and as it dips into the T tubules, it causes depolarization. This depolarization is sensed by dihydropyridine receptors. The dihydropyridine receptors in turn activate the Rhinodine receptors, which are the calcium release channels present in the terminal cisternae. So the calcium ions are released into the sarcoplasm. The calcium ions then bind with troponin C. As the calcium ions bind with the troponin C, the tropomyosin strand moves deeper into the groove between the two F-actin strands, exposing the active site of actin filaments. The head of the myosin is already ready to bind to the active site of actin filaments, but in the resting state it cannot do so because they are covered by the tropomyosin. So when calcium combines with troponin C, the tropomyosin strands move deeper into the groove, exposing the active sites, and therefore the head of the myosin combine with the active sites of actin filament. Immediately after combination, there is a power stroke which causes dragging of the actin filament towards the center of the myosin filament. Then the head of the myosin detaches from the active site because of the attachment of ATP. The ATP is broken down, the energy used for the recovery stroke, so that now the head of the myosin goes back to become perpendicular again, comes in contact with the subsequent active site of the actin filament, and the same processes continue, that is the power stroke, detachment, recovery stroke, attachment, power stroke, detachment, like that, and this cross bridge cycling continues, and that is how the actin filaments slide towards the center of the myosin filaments, and that is the process of contraction. So this is a summary of the same. As the depolarization wave passes along the T-tubule membrane, it activates the DHP receptors, which in turn activates the Rhinodine receptors through the junctional feet in the terminal cisternae, and since these are the calcium release channels, the calcium channels open and they release calcium ions into the sarcoplasm. As the sarcoplasmic reticulum surrounds the myofibrils, the calcium ions released from these calcium release channels have only a short distance to diffuse to reach the troponin that is present in the actin filaments, where its binding initiates the process of contraction. So, after considering all this, what do you think is the factor that couples the process of excitation and contraction? The answer is simple. The coupling agent is the calcium ions. So remember, the coupling agent is the calcium ions. Excitation of a muscle ultimately brings about release of calcium, and then the binding of the calcium with the troponin initiates the process of muscle contraction. So the excitation contraction coupler or the coupling agent is the calcium ions. Right. The same here. This is as the depolarization spreads along the T-tubules, it activates the dihydropyridine receptors, which is a voltage sensor, and the dihydropyridine receptors in turn activates the Rhinodine receptor, which is a calcium release channel present in the terminal cisternae, and therefore when this channel opens, the calcium ions enter the sarcoplasm and initiate the process of contraction by combining with troponin C, and once there is a repolarization and the resting membrane potential is restored, there is no more activation of DHP receptors, so there is no more opening of these Rhinodine receptors or the release channels, and then the calcium ATPase that is present in the sarcoplasm is activated. It pumps the calcium ions back into the L tubules where it binds with the calsequestrin so that more and more calcium ions can be stored, and once the calcium concentration in the sarcoplasm falls, the calcium from troponin C dissociates from the troponin, and once it dissociates from troponin, the tropomyosin is no more tugged, it automatically comes back on the active sites of actin filament, and thus there is no more interaction between actin and myosin. Alright, the same thing mentioned briefly here. As you can see, the action potential going along the sarcolemma dips through the T tubules. These are the L tubules, the calcium release channels which release the calcium ions. The calcium ions then bind with the troponin C, opening the active sites so that the head of the myosin react with them. There is a cross bridge cycling, and finally the contraction. Relaxation of muscle takes place by removing the calcium ions from troponin, achieved by decreasing the calcium concentration in the sarcoplasm. So remember, relaxation of a muscle is also an active process. It needs ATP. ATP for what? The membrane of the sarcoplasmic reticulum contains a carrier mediated primary active transport system, which I mentioned already, that is the calcium pump or the calcium ATPase, that pumps the calcium ions back into the reticulum. The calcium is rapidly released from the reticulum upon arrival of action potential, but the pumping of released calcium back into the reticulum requires a much longer time, and therefore the contractile activity continues for some time after the action potential. In other words, the mechanical activity exceeds the duration of the electrical activity that occurs. So remember, just as contraction is an active process and requires ATP for the cross-bridge cycling, the relaxation of skeletal muscle is also an active process and requires ATP for the action of calcium ATPase or the calcium pump that is present in the wall of the L tubules, which is pumping the calcium ions back into the L tubules, lowering the sarcoplasmic calcium concentration and thereby initiating relaxation. So that was about the excitation, contraction, coupling.